Hello, everyone. Thank you for joining this installment of Boyce Thompson Institute's new Breaking Ground discussion series. My name is AJ Bushy, and I'm in the Communications Department here at BTI. And for this month's Breaking Ground, we welcome Dr. Lukas Mueller, bioinformaticist here at BTI, who specializes in database architecture and plant breeding. And today we'll be talking about some of the plant breeding databases that he's helped develop and create, and how researchers, plant breeders, and farmers around the world are using them to improve crops, with a focus on crops that are important to food insecure regions. Hi, Lukas.
Good morning, everybody.
Good morning. And first, a few quick housekeeping announcements. During the session, all participants will be muted, and questions may be asked in the chat, and a Q&A session will be at the end of the conversation. And it's usually about a half-hour discussion, then a half-hour chat with everyone else. And you'll see the chat box. I'm sure you all know Zoom by now. There's a chat box and a Q&A box. You can throw stuff in either one. We'll get it. And I also want to thank Holt Architects for sponsoring today's Breaking Ground discussion. So thank you to those folks. And thank you all again for joining us today. It looks like we got a good batch of folks. We got 40 people out there, so that's awesome. So thank you for joining us. And to start off with, Lukas, could you tell us a bit about yourselves?
Yeah, hi. So good morning, everybody. My name is Lukas Mueller, and I'm a professor at the Boyce Thompson Institute. My field is bioinformatics and databases. And so how did I get here? Well, I'm actually originally from Switzerland. I did my undergraduate degree in the Swiss Federal Institute of Technology in Zurich, and then did a PhD at the University of Lausanne in plant biochemistry. So I'm actually a biochemist by training, and then did also a postdoc in the US in that field, but switched at that point to databases because in the lab next door, they created a new kind of database called TARE, or arabidopsis.org. And I was very interested in that because I had a lifelong interest in computers. And that seemed to be a really great way to combine my interest in computers and biology. I have to say that doing research, active research in biochemistry in the lab, such as I did in the Walbot lab at Stanford, that was one of the best time of my life. But I think I could really combine my interest in computers and biology, and that is just opened a new amazing field. And then from there, I went to Cornell with Stephen Tanksley and started running the soulgenomics.net website, which is a website about tomato biology and related solanaceae plants such as potato and petunia, tobacco, peppers, and so on, eggplants. And that has evolved later into this field of breeding databases that we're going to talk about today, I guess. And so this was actually a big step, to go from a genome databases such as SGN to breeding databases, and it was really I really got into this because I maybe shouldn't tell this story, but it was a story about how networking is important in research, and my son had soccer practice every afternoon or something. No, I think it was once or twice a week. And I had to sneak out at 3 p.m. and had a really bad conscience about that. And so, but there at the soccer practice, there was this other dad who had also his son at soccer practice. And his name is Jean-Luc Yannick. And we started talking. Of course, I knew him, from the faculty at Cornell, but I didn't really know him that well. And so we started talking, and he said that he was just writing this grant about Cassava, which ended up being the next gen Cassava grant, which is really his brainchild. And also, I think, a visionary program manager at Gates. Her name is Kathy Cann. So they really were, it's really their brainchild. And so they asked me to participate in that grant, and that's how Cassava base came along. And so we expanded from cassava to other root and tuber crops and banana, which is a section of the CGIAR breeding activities. And now we run databases for, I think we have it on the slide, right? Yeah, exactly. So we created cassava base, and then we had, now we have database about sweet potato, banana, which is called musa base, and yam, which is called yam base. And you can see that this really came from a database called Soil Genomics Network. And also this spawned a lot of other genomic databases, such as citrusgreening.org and fernbase, rhizomum.org and milkweed base. And these last three ones are actually collaborations with other BTI faculty. And so what we try to do, and this is shown on the left side of the picture, is breeding informatics was, for some reason, it's actually something quite hard, because it's a very complex process, right? And it involves phenotyping and field layouts and statistics and so on. So it's actually a fairly complex process. And what people have done, traditionally, they've just used Excel sheets, and they kept all their data in Excel sheets. And that works quite well if you have a small breeding program, if you don't have multiple sites. But the Next Gen Cassava program was a huge program, with sites on several continents and many breeding centers involved. And so we needed a better solution than Excel sheets. And so what we try to do is what we call the digital ecosystem. And I'm not going through this whole graph here, but the central idea is that you keep all your data in a digital format, and you never type something in an Excel sheet, because the Excel sheets are good, but they're very hard to compute with. They're very hard to manage. They're very hard for data aggregation is very cumbersome and error prone. And also the other goal of the Next Gen project was actually to use genomic selection, which really uses genotypic information to get at breeders' decisions, right? So normally in the old days, breeders would just phenotype, and based on the phenotypes, they would create new crosses. But that's, yeah.
So just real quick, so when you say phenotype, you mean like a physical characteristic, so like a large sweet potato
exactly.
versus a small one?
Yeah, that's what that means. You see a plant with big tomatoes, you might want to cross it with another plant with big tomatoes, and you get even bigger tomatoes, right? But there are actually a lot of traits. So we call these traits. The tomato size would be a trait, right? The fruit size. And there are actually a lot of traits that are not easy to phenotype visually in the field. So for lots of phenotypes, you want to use markers, genetic markers, such as for disease resistance. But then there are a group of traits that are very hard to even genotype. There's no marker for them, like yield. Thousands of genes are involved in yield in that trait. So you cannot genotype for it. But what you can do is you can genotype the entire genome and calculate for every marker, and there are tens of thousands of markers in the genome, how much it contributes to yield, because you can do that with different accessions that have different yields, and you can build these correlations. And then if given a genotype, you can actually predict what that genotype would yield. And
that's you could use. It seems like you could use this to discover new markers. Is that correct?
Yes, you can discover new markers, but here you actually use all the markers in aggregate. But yeah, sometimes there will be a marker that maybe you can discover a marker that has a huge contribution, and then you could just use that again, like with the older model. You could just use that marker as a analytical tool. like we have for a lot of disease resistance genes and so on.
Okay.
And so one of the really interesting things is when you do this is that you can actually accelerate the breeding cycle by a huge factor, because genotyping is actually fairly fast. You just have to extract DNA, send it to a DNA sequencing center, process the data, load it into the database, and this can be done in a matter of weeks. Whereas if you have to grow out a plant and phenotype, that can be a matter of months or even years, right? And so you can actually achieve a huge speed up of the breeding cycle. And it's like with a bank account where you get compound interest, right? If you select more, if you shortened your banking years and you got the same interest every month, that would be, your money would accumulate much faster. And of course, the yield accumulates much faster as well if you do that for plants.
And that's particularly important for cassava, right?
Yeah, and exactly. So some of these plants we're working with, some of these crops, they had huge cycles for breeding. They would take between 5 and 10 years. And with this technology, you can bring it down to 1 to 2 years. So that's approximately five times increase, which is amazing.
Yeah. So what else is there about cassava? Like, you mentioned next gen cassava and the cassava base. Could you talk a little bit more about how this the cassava base can help?
Yeah. So first, maybe a little bit about cassava, because I think many people don't know how important it is. People know that rice and wheat and corn is important, and people, especially in the West, are not familiar with cassava at all. And I have to say, I wasn't familiar with it either. I'm a biochemist by training, so I knew about the secondary metabolites of cassava, but I didn't really know how important it was for nutrition in many parts of the world, especially the tropical parts, of course. And Africa today, I think, is still the #1 producer with I think 2/3 of all cassava production, half to 2/3, I think, somewhere in that region. So it is hugely important in Africa, and there are hundreds of millions of people who depend on it every day. So cassava has not received a lot of attention from breeding companies or from breeders in general. There are a number of breeding centers that work on it, the CIAT in South America and the IITA in Africa, along with national programs. But of course, that's a relatively small effort compared to, if you take something like corn, that has received such huge input into its breeding, and in many presentations by corn breeders, you will see this famous curve of how corn yield has increased over the years, and it's just astounding, right? What has been done with corn, and so the idea is that with cassava, we could probably pretty easy, get a lot of gains with just a little bit more input into the breeding pipeline, and especially using these modern approaches that are also being used on corn, that would speed it up a lot. So cassava is actually eaten in West and East Africa in the tropical regions, and I think here we see cassava at a market in Nigeria, and how it is eaten mainly in West Africa is as a dish called gari, and so the cassava is ground, and then cassava is heated up, and that is done because I think cassava is maybe quite unique among food crops in that it is toxic, and that it accumulates cyanogenic, so-called cyanogenic glucosides. Cyanogenic glucosides. Sorry, it's a hard word to say. And essentially, what that does, it's a secondary metabolite. And when you crush the cell, it will release cyanide, which is of course very toxic. And so cassava has to be prepared in a special way so that it's safe for consumption. And what people do is they have these, you can heat it or you can boil it, and that will make the cyanide evaporate. So that is very important. And so actually, a graduate student who works in my lab at the BTI has recently analyzed using genetic and genomics methods, what the important factors are in cyanide content in cassavas. And he actually has isolated a new gene, or identified a gene, I have to say, from the genome, that is apparently the gene that makes the difference in these lines. And surprisingly, it's actually not a biochemical gene. There are genes that are responsible for the biochemistry, for the enzymes that essentially make this compound, but it's a transport gene, and that's very interesting. That would be the mechanism by which cassava modulates cyanogen content. Cyanide.
It seems interesting. An exporter gene, is that?
Yes,
that does seem interesting.
Something like that. It's a membrane transporter.
Transporter, and
yeah. And Alex Ogbonna is the person who works on that, and it's really great work, and it's just been accepted for publication, so that's always exciting.
Very cool. Congratulations, Alex.
Yeah. Actually, he's here in the field. You can see him and Guillaume Baucher, who was a researcher as well in the group. Another big problem with cassava is that it's susceptible to some viruses, and especially in East Africa, there is a very high prevalence of this cassava brown spot disease, CBSD, and that's really important to find lines that are resistant to it, because it really can destroy your crop to a large extent. So you see these brown spots that appear in the root, and I have to say the root is the part that is eaten. I think I haven't said that yet. So it's a root crop, and of course roots that look like this are not good for consumption anymore. If it's even like this, I think you shouldn't eat them anymore. So it's a very difficult problem for cassava, but I guess we are working on it, and there are resistant lines now.
Is that caused by a virus or a bacteria or a fungus?
Yeah, so this is caused by a virus. So viruses are not, humans are not the only organism who is suffering from viruses. Cassava is too. So yeah, it's very important. There's also the cassava version of social distancing and so on, because you don't want to get these viruses out in the field. So you don't really send cassava material across borders and stuff like that. So it works a little bit the same way as with humans.
But they do make seeds, right? Can you pass seeds across border? Like, how does and I guess that comes to a bigger question of mine, that it seems like, the cassava base, it would work. It seems like it would need to be somehow tied to the actual sample. So if you discover, like
exactly,
you mentioned,
yeah, that's an important point. Yeah.
If you find a strain that's disease resistant, how do you get those seeds to other countries? So you'd have to tie it back to a physical specimen somehow.
Right, exactly. So seed is much safer than other types of tissues, and of course, you like with humans, there's quarantines and so on. So you can actually quarantine, or you grow them in a place where you don't threaten any other cassava production, and then you can decide, is it virus free? If so, after a quarantine, it can go on and be used in other breeding centers. But it's a complex process, and it's kind of it's complex and long. So it's not that straightforward to send seed around around the
Right.
world. Yeah. Surprisingly, it's much easier to send data around.
Right.
But that's not always, the solution to your question, because you sometimes do need, as you say, in the end you want to grow something and you do need the seed. So cassava is actually a clonally propagated. So farmers don't actually grow it from seed; they grow it from stem.
Mm-hmm.
And so that's the stem, of course, will contain virus, and you can spread the virus with stems. So we have to be very careful. But it is a clonally propagated crop, and also what that does, or what has happened over the years, is that when you clonally propagate something, they lose the ability or the desire, I want to say, to make flowers. And it's actually it was a huge problem in cassava, that they actually don't flower that much. So there was actually a researcher in the Next Gen Cassava project here at Cornell, Tim Setter, who looked at that question, and he found methods to increase the flowering in cassava. And also, I think the CIAT in Colombia, the research center in Colombia that works with cassava, they also helped a lot to answer that question.
It's interesting, because it didn't need to anymore. It just kind of evolved to.
Yeah, exactly. It has to do with plant hormones and so on, and and also they found that when you illuminate fields with red light in the evening, that increases flowering a lot, because obviously red light detection is a very important developmental signal for plants. Cool. So I thought those were quite interesting approaches, and I think quite innovative.
Yeah. Okay, very fascinating. Let's move on to sweet potatoes. That was another one that you mentioned, and that has some similarities to cassava, some differences as well.
Yeah, it's also a root crop. I think it is, in terms of calories for people, it's not as important as cassava, if I can say that way. It's still very important, but it's also, I think, important for nutrition, not only calories, because sweet potatoes, especially if you eat them in the US, they're usually orange fleshed or yellow fleshed. In Africa, I think the white fleshed sweet potatoes were much more common. And not our project, another project called Sasha has actually introduced the orange and yellow fleshed varieties in Africa. And I think they save a lot of lives with that, because the vitamin A concentration is much higher there, and it really prevents vitamin A deficiency in people. The people who did this won actually the World Food Prize I think in 2017. And I think they're here on the slide. Say again?
2016, I believe.
2016. Yeah, sorry, I might have the year wrong. And these were people working with the Sasha project. So these are the winners were Maria Andrade, Robert Manga, and Jen Lowe. And we work with these people too. And so it's of course huge honor, huge pleasure to work with that caliber of people. Also, another person working on that is Craig Yencher from NCSU, who is a huge sweet potato expert, and he also has an interesting sideline. He does also ornamental sweet potatoes, which is kind of interesting. But here on this slide, on the left side, you see sweet potatoes, how they come out when they are harvested, and you see that they have these barcode on them, and that's the direct link to the database, right? So that tells, when you scan that barcode, you would know everything about that sweet potato, where it was grown, what line it is, and so on. So what genome it has potentially. What phenotypic information has been scored on it, and so on.
And so it's interesting you you mentioned these folks who won the World Food Prize, and how there's almost kind of like a social hurdle of getting people to eat the orange or yellow flesh sweet potato, and when folks weren't used to it, and how important that was for the vitamin A. I think
I think it's actually a huge problem, right? That when you make food different, then people, the acceptance by people might not be there, even if it's healthier. And imagine, if it happened here, if something that you're used to suddenly is different, right? Then people might not like it anymore, although it's just different, it just looks a little different. And so these social hurdles are really important. And all these projects have a lot of social scientists in them as well, because they study kind of these questions. No, in a breeding program, it's always an interesting question. What do you breed for, right? I mean, you could breed for anything really, and you probably would get there. But what makes sense to breed? And of course, there's some easy, or clear targets like disease resistance. You need to have things that grow in the field. But then, consumer acceptance, and what does the customer want? What does the farmer want, right? These are all questions that are important too. Because if the farmer doesn't want to grow it, nobody will eat it, right? And if people don't want to buy it because they don't like the taste, or don't like how it looks, then you're not successful either. So that's why there's a lot of on-farm trials, and now we are starting to collaborate with another Gates project called RTB Foods, which really looks at consumer preference. Because I think there have been a lot of breeding products coming out of the breeding centers, and the acceptance was low. Maybe also the quality was low in the sense that they didn't pay attention to the cookie quality, or maybe just what it looks like, or so these things are all analyzed by the RTB Foods program, and it's a very important project to make sure that that you actually breed for the right traits.
Yeah, and that's always struck me about a lot of your work is this collaborative nature, and how you're putting together some infrastructure that other people can use and figure out what they need. So that it's the farmer, it's the breeder, it's the local farmers, the local breeders, the local people who are deciding what they need, and using the data to create what they need and discover the things they need and develop the crops that they need. And I always like that aspect of your work, the collaborative nature of it. I think that's great.
Yeah. I think that's actually one of the exciting aspects. That's why I like to do this work, because it's so collaborative. I guess when I used to be on the biochemistry side, it's more kind of, you're more the lonely fighter that wants to make a discovery or something. and these projects are huge, and they bring people together from the social sciences, from informatics, from genetics, breeders, really from a lot of different directions, and it's hugely interesting. It's hugely enriching to work with all these people. I find it amazing. Yeah,
it's cool. And one other interesting thing about sweet potato, I understand, is that it's difficult to work with genetically, because it has 6 copies of each chromosome, whereas we just have 2 copies of each chromosome. It has 6, so that's going to be very tricky to tease out. If you have 6 different alleles of a chromosome, like what's doing what in there, that's going to be.
Exactly. Yeah, it's a hexaploid, and so it has 6 chromosome copies instead of just 2, like cassava, and that makes breeding much more difficult. Also, genomic approaches are much harder, of course, because you have way more data, and it's harder to assign even what does this marker mean, and how many copies are there exactly, and so on. That's all things that become much harder to do. But we're working on it, and actually currently there's only the genome sequence of diploid sweet potatoes, but a lab at BTI actually works it's part of this project actually, works on the hexaploid genome as well. And that's Zhang Fei's lab who works on this, and I think they're actually quite far along already. I think it will be released soon if it hasn't already been released.
Okay, let's we're getting close to a half hour. I also wanted to talk with you about bananas. So I know you have the Musa base or Musa base.
Yeah, Musa base.
Musa base.
Yeah, so banana is, how should I say that? It's a mess. It's very, very difficult to breed bananas. It's I think the hardest. I mean, we have seen sweet potato is hard. There's certain aspects about yam that we haven't really talked about is hard. But bananas are super hard because if you eat the banana, you realize you're eating the fruit, but there are actually no seed in the fruit, right? No seeds, right? So how can you do breeding without seeds? That's impossible, right? So why are there no seeds? Is because these bananas are triploids, and the triploids don't make seeds. There are other bananas that are diploids, and they also don't make seeds for other reasons. Because you cannot really eat the banana that makes seeds. The seeds are actually very hard and large and fill the entire banana. So it's very, very hard to have seed generating bananas and to sell them and eat them. That's not something that works. So how they do it is they have diploid bananas and tetraploid bananas who can generate seed, and those are then crossed and in a weird Frankensteinian way create the triploid bananas, and those are then clonally propagated. So they're purely clonally propagated. And so everybody might know the Cavendish banana that we buy at the supermarket here, and that's actually a clone, right? And you've probably read in the newspapers that that's a huge problem because it's exactly identical plant all over the world. They're really clonal, right? So if there is a pathogen that can infect the banana, it can infect every plant in the world, basically, right? Which is not true for cassava lines because they're all genetically a little bit different, or many other crops. But when it's really truly a clone, yeah, it's over. As soon as there is a pathogen that overcomes the resistance in that strain, it's over. And this has happened actually in the 1950s with bananas. And the line that was prevalent then, called Gros Michel, had to be replaced by the Cavendish line, which was then resistant to the Panama disease, as it's called. And now we have, I think, strain 4 of the same pathogen has overcome the resistance in Cavendish.
Oh, wow.
And it will actually probably wipe out that strain completely for market purposes, for large scale production at some point.
So that's the Cavendish banana, but that's different from some of the strains that are eaten in like Eastern and Central Africa, for example, correct?
Yeah, exactly. That's exactly correct. So it's not as bad in these cooking bananas. They're starchy bananas. They're not dessert bananas like the Cavendish. And they are genetically much richer. There's much more variation locally and in the breeding programs, and so they are not as susceptible to this threat of one pathogen comes along and wipes out everything. But it is with every clone club, of course, a little bit more of a concern, and so. Yeah, so these bananas are actually quite different. You have to cook them. They have very nice meals that they make out of the bananas in Uganda, a very famous recipe is called matoke, and I think the bananas really are also called matoke bananas because of that. You know, they made like a stew out of the bananas, and they boil it in a banana leaf, and it's extremely flavorful and nice. I really love matoke. And in Tanzania, there are actually different bananas. There are mchare bananas, and they have a completely different cuisine associated with them. They're just boiled and then eaten with a sauce. And it's also very good.
Yeah, they all sound good.
I'm a big matoke fan.
Okay, let's, I know you had an acknowledgement slide. So if we could maybe show that, and then we can move on to some Q&A. Looks like your lab is there.
Yeah, so this is my lab. This includes a lot of visitors as well, and undergrads. So the lab isn't quite that big, really.
That's a lot of people.
It's a lot of people here on the slide. Yeah, I think one of the amazing things is in working, you know, we talked about working with all these people in the breeding centers and the social scientists and the, you know, the geneticists and so on. But of course, it's also great to work with all these people in the lab who are just amazing. I mean, it's really a great thing to work, to have an amazing team, and so happy to be working with them every day. So
it's great. Very prolific.
Yeah.
Okay. I don't see any questions yet. There's one comment. I hope the next banana clone actually tastes like something.
Yeah, it's a good comment because, you know, we are used to Cavendish. And when the Cavendish banana came along, people said, well, it has completely lost all flavor, right? And the Gros Michel banana actually tastes quite differently. And I've tasted a Gros Michel once in Africa in a field. They grew it, but I'm not sure if I detected a huge difference. But I heard that, you know, this artificial banana flavoring that you can buy, that doesn't really taste like banana, but that Gros Michel had more of that flavor. So it was more of an accurate flavor for Gros Michel than for Cavendish.
Interesting. Okay. Yeah, I always hear stories about how that old banana tasted better than the current banana, but I have no idea.
Yeah, I guess it's like tomatoes. You know, the tomatoes used to have flavor too, and through, you know, because the truth, I mean, it is, I think, a problem in breeding, that people focus a lot on yield, right? And whatever you focus on, that's what you get in breeding, right? But everything you don't focus on, you actually lose, right? So if you don't focus on flavor, you will not have flavor. If you don't focus on shelf life, you won't have shelf life, right? I mean, you can focus on anything, but people, I think breeding programs really always put a priority on yield, because that's what in the end, you know, gets you the money, right? And makes the farmers interested in actually growing it, because the farmer maybe wants to grow a super flavorful tomato, but that has a very poor yield, and you can't sell a lot, right? So in every chain of that process, you need to have the right traits.
Well, that's where your databases come in, right? So then you can find the disease resistance genes and the flavor genes and the yield genes, and try to get them all into a single
right
plant or fruit, whatever seed. So I don't see any other questions yet. You've mentioned yam a few times. Do you want to talk a little bit about yam with our extra time?
Yeah, yam in a way, yeah, yam is also grown mostly in West Africa. So places like Nigeria, Ghana, Cote d'Ivoire, and it's actually a very interesting plant. And one thing I find really interesting is that sweet potatoes are sometimes called yams in the US. So you go to
That's
the
true.
store, you can buy yams, but you're actually buying a sweet potato. But a real yam is actually something completely different from a sweet potato. It's actually, a sweet potato is a dicot. Yams are actually monocots.
Oh, really?
So kind of, yeah, biologically speaking, in terms of plants, they're as far apart, phylogenetically as they could be.
So it's more closely related to things like grass and rice
Yeah,
and corn
exactly.
and those monocots, right?
Yeah. And so every plant has a little, a little detail that makes it a little harder to do breeding. And we have talked about sweet potato. We have the hexaploid. Bananas are super difficult for many reasons. And yams have this this feature that they are dioecious, and that means that they're female and male plants. So there's always this for the data management side, right? You need to track these things because you cannot, if you want to do crosses, you need to be aware of whether the plants are male or female, whereas you don't have to do that in cassava, right? So that's an interesting little thing about yams.
We do have one question in from Greg Traxler. How do you evaluate progress in a crop improvement program? What metrics do you look at? Reduce cycle time, etc. So does that depend on the crop, or?
Yeah, that's a good question because, what you really want to see is you want to measure the gain in yield. That's what, unfortunately, that's what people consider progress. Okay. And of course, disease resistance obviously feeds into indirectly also, of course, into yield loss, right? When you don't have a resistant plant, and so I think the yield is a good measure of progress in some ways, but it's not the only way. I mean, and I think that's traditionally how it has been done. We want. I think the Gates Foundation wants to see 1.5% gain every year, right? But I think in the past few years, there's been a realization that, yeah, that's great, but now we have this super high yielding plant, but farmers in the field, they still grow varieties that are 20, 30 years old, right? And so it has basically no impact in the real world if we have essentially some lab strain that has a super yield, right? So how do you get it out there? And I think that's going to be the new measure and the new way to kind of judge a breeding program. Do they get their good clones out there to the farmer? And that's something that has to be measured and has to be documented, and you have to be able to show that your breeding program has a real life impact and not just some.
That's interesting. That kind of ties into another question we just got. How are farmers in remote regions of Africa benefiting from databases like Cassava Base? And that's kind of sounds like we still need to measure that and figure that out.
Yeah, I think the idea here is that a database like Cassava Base is really, currently at least, kind of the target audience of the database is really the breeding programs, and we are not really addressing the farmers at this point. But what we would actually like to do is we'd like to add new features where, because many of the lines that we have in the database, there are 400,000 cassava lines now. They're mostly very short-lived intermediates in a breeding program that you cannot order anywhere, or you cannot use them in the field. So a feature that we would like to implement is the idea of a seed catalog that you could directly, the breeders can directly elevate some of the accessions in the database into a catalog that farmers could browse, and they could see the features of all the recommended, or the lines that are around for long term that you can actually order, right? And they can browse them and they can decide for themselves which ones they would like to order.
Great idea.
So I think in soon, I think Cassava base might be a portal for farmers in the field to directly check out what's new, you know, what's there, and you can benefit directly.
Okay, we have another question from Nishit Nandankar, if I pronounced it right. I'm curious about database development. Did you entirely build a new database software, or is there a particular database software that you would recommend for breeding programs that are trying to develop resources for other crops?
Yeah, so we built this database mainly on the soil genomics side. So we have developed the database in a way from scratch, but in the process, we have actually talked to other databases, and we have co-developed this database schema called the Chado ND schema, and that's what our database is based on, and other databases as well. And so there are a few other databases out there. When we started to do Cassava base, I think we were the only one that was really completely open source and completely web-based. But a lot of other databases are moving towards that model now as well. So there are a number of databases now that you could use for your breeding program. But Cassava base or breed base is completely open source. You can just download it and use it if you want to. Of course, we'd love to help manage your data, and I think that's really what we are doing. We are in data management, and so of course the one I would recommend is Breedbase, but there are a few others out there that you probably should look at.
And that is what you just published on that in PLOS One, right? Was that just like Monday or last week?
Which one?
The Chado schema of Breedbase. Isn't that what you just published in PLOS?
Oh yeah, that's a good point. So
can I put that link in the chat? Is that all right?
Yeah, sure. Yeah. So we just published a paper about one aspect of how we store genotypic information in the database. And I think it's quite a nice way of doing it. It's very simple. It's integrated into Postgres. It uses something called JSONB. It's a great way of storing that kind of information in the database, where you have lots of data points that you cannot store individually as as a database row. You can use these other types that Postgres manages really well. So yeah, look at that paper. That's I think it's quite good.
Okay, I'm just going to go down.
I just want to mention it's Nick Morales implemented that. He's a PhD student in Cornell and my lab, and yeah, I think it's an amazing job. Yeah.
Yeah, he's the first author on that paper. So.
Yeah.
We have another one from Vijay Joshi. 2 quick questions. Would the Veritome database incorporate proteomics data at some point? And are there plans to have a similar database for other members of the Solanaceae species?
Well, so Veritome is actually a specific project that was funded by the NSF and in which we served as the data portal on soilgenomics.net. And we can integrate quite a lot of different types of data. I don't think we have planned to integrate proteomics data with that data set yet, but I think it could be done. And what was the other question?
Are there plans to have a similar database for other members of the Solanaceae species?
Yes, I mean, we would love to. We can generate these databases. I mean, the database essentially exists. We just need to fill in the data and have a tweak here and there, because every project has its own little idiosyncrasies, right? So you have to make little adaptations often. And yeah, if you wanted to add proteomics data, you would have to. I mean, I think our platform could could accommodate it because it's very similar to other types of data that we already accommodate. So yeah, we would love to do it, but I think we would need a project and funding for it.
Yeah. Speaking of funding, the next question is, how are these databases funded and how can they be sustained after the current funding lapses? And could it be crowdfunded?
Yeah, that's a good question. I don't have the complete answer to it either, I guess. The database is, one second.
It's okay.
The database are funded by the Bill and Melinda Gates Foundation. And there are sometimes other funding partners, such as the British Development Agency as well, funds part of Cassava base. And they have provided, so we are in the second cycle for most funding cycle for most databases. Hope there will be a third one. But it's clear that, and that was the initial plan, that these databases at some point need to be moving. Now we are running them at PTI, and you've seen, I think on the first slide, there was the server farm that we have here. And at some point, they need to be moving to the actual breeding centers. But that may not happen for another, maybe after the next cycle, I think. As the software becomes more mature and we have made a lot of progress in how to install the databases, so you can easily install them using Docker. It's essentially one Docker command, and you have all the software that you need. But the big problem is, of course, that the software is actually not the problem. I think in many ways, the problem is organizing the data. That's the big challenge. And when you have a database, you need a very systematic way of naming your germplasm, for example. And for many programs don't really have that. And so you need to probably go through and rename a lot of your germplasm. And if you have thousands of germplasm already, that's a huge amount of curation work that you need to do before you can even start thinking about a database. The other thing is traits. What do you measure and how do you measure it? And we're using so-called trait ontologies, and this needs to be very clearly defined what exactly you're measuring, because especially, for small breeding programs, it's not that important. So if you're just one location or a few locations, one breeding program, you have a lot of freedom what you do with naming. But if you're a larger organization or a larger project, it becomes hugely important how to do that, because otherwise, you end up with data, and if it's not uniformly been measured, you cannot compare the data, and then kind of the database becomes a little bit moot, because wow, why would you put data in the database that you can't even compare to other data in the database, right? That's the power of the database, that you'll be able to combine a lot of data that you could never combine before, because the data was on somebody's Excel sheet on a laptop, and who knows, nobody knows what's there. Here, everybody can see what's there, and you can say, well, that's interesting data to me. I will go and ask. And we always say you have to ask, though, right? You don't just download it. And you can collaborate with people much easier.
We're coming up to 1 o'clock, so I'm just going to do one last quick question. Hopefully it's quick. We'll see. I'm going to paraphrase. So in discussions you have with programs that you work with, how do you work to prioritize traits other than yield, and how have you incorporated the input from the social scientists?
Yeah, so that's a good question. Of course, we don't do that as the database. That's the breeders, and the whole project does that. We're just trying to kind of organize this information, what they come up with. But of course, I've been in a lot of discussions. I remember the first time I went to a Cassava meeting, and the breeders were talking about traits. And I thought, well, that's useful. You need to know what you measure. Then the next year we meet again, and I thought that subject was now closed, that we know what we're measuring now, but they were still just talking about traits. And so that's something that you always talk about, right? The breeders, that's their main thing. What should we breed for? That's the biggest question, right? And so they constantly talk about it, and they constantly prioritize it, and they take all the data that they can to do that, like from the social scientists, from the quality labs, from the genotyping data, and so on.
So is it easy to add new like trait data into the database? I'm sure I worded that right. Like, is it just like add a new column, like in the Excel sheet kind of thing? And
in a way, it's like that. It's a little more complicated, but it's all done in the back. You just say, I want this trait, and it magically appears in the database, and you can use it. Yes, but you have to tell. We have actually a page on the database when you need a new trait, and that happens a lot. That people want to measure something new because there's a new problem or there's a new quality control thing that you would like to measure. And there's a web page you can go and say, I want this new trait, and then it will be incorporated into the ontology and it will appear on the site. But of course, we don't want everybody just to add traits, because then we would have, you end up with duplicated traits and so on. So it's important that a curator looks at all these terms, that knows all the traits that are already there, knows where to put these traits in the ontology, which is a hierarchical structure. And so it's a very, it's kind of labor intensive, and you have to have a lot of knowledge about that species, that crop, and that ontology.
Okay. We have just 2 more questions. If you're okay with staying a little, another 5 minutes or so.
Sure.
The last 2 questions. All right, if folks want to stay, they're good questions. Here's one from AC Rupka. I'm gathering that Cassava base is a shared resource between multiple Cassava breeding groups. Are there any concerns around proprietary data sharing?
That's also a very good question, because I remember initially the breeders were extremely concerned that they had to, that essentially all the data is out there, for everybody to see in the entire world, right? And that was a very new concept for them, because before, as I said, in the Excel sheet on their laptop, nobody could see the data. And I think there has been a huge shift in mindset, about data and data proprietariness. And I think now they're totally comfortable with it, sharing it. And I see when we start with a new crop or a new breeding program, and of course, if you run the database yourself, if you download BreedBase and run it, you can run it however you want it. You don't have to make it available to everybody in the world. But these Gates Foundation grants, they like to see that you make data available to everybody. And so we do that. And I think after a little bit of resistance and sometimes a lot of resistance, people are actually kind of happy with it, because it gets their data out there. Other breeders see it. They might see mistakes in the data. They might see patterns in the data. They might have ideas for collaborations and so on. So I think for these kinds of crops that are not really, there are no breeding companies really breeding for them. So there's really not much money to be made in keeping the data private. It's better to get it out there.
Okay, we'll do one last question. Do you have any examples of where the databases brought a community of users together, or is it more typical for the community to get together and build a database? That's a good question.
Yeah, that's a good, an interesting way of putting it. I'm thinking that in many ways, I think the database brings a lot of people together, because the breeding was a little bit of an insular activity. Every breeder ran their program with their Excel sheets, as I say, and this is really, I think it has really brought people together. And I mean, it's not just a database, of course, it's these projects in a larger sense, that bring people together. And people, I feel that this makes a huge difference and has a huge impact in the way that breeding is done and in the efficiency in which it is done. So I think the database is a component in that, but yeah, that bringing these people together is hugely important. And that's really, the vision of the Gates Foundation and the wisdom.
It sounds like, especially for the crops we discussed today, as you said, they're grown on 3 continents, at least, right? Africa, South America, and North America, the Caribbean, all over. So that'd probably be pretty hard to do without the new virtual world.
Yeah, exactly. I mean, it would be very difficult to run breeding programs without a central web-based database. I mean, another big, why people hadn't developed web-based databases before is that, of course, the internet has to be pretty good for people to be able to use it. And if you go to certain faraway places, the internet becomes much less good.
Yeah.
But it turns out that right now, I think it's totally usable from wherever our users are, and it only gets better. I don't think it will get worse.
Hopefully. Okay, well, we'll call it there. So thank you again, Dr. Lukas Mueller, for taking the time today to chat with us about your work that you're doing. And thank you to everyone out there who joined us for this new series of virtual chats. And I just want to let you all know that you can read more about BTI's current research and many other neat stories about BTI science in our 2019 annual report, which you can find online at btiscience.org/annual report. And it's really neat. It's all virtual, all online last year for the first time, and there's links to videos and all kinds of neat stuff in there. So I hope you check it out. And also I ask you all if you could please join us for BTI's next Breaking Ground discussion series, which will feature an interactive chat with Boyce Thompson Institute's Maria Harrison, and that will be on Tuesday, December 15th. And we'll be talking about how the microscopic relationships between plant roots and soil fungi could help ultimately reduce fertilizer use and the pollution that comes from too much fertilizer use. And just to let you know, Dr. Harrison, she takes tons of neat pictures with all kinds of cool microscopes. So it's going to be very visual, lots of neat pictures, movies, animations. It's going to be a very visual Breaking Ground. So I hope you join us for that. And you can go to btiscience.org for more information and to register for that. And thanks again for joining us. And I just want to reiterate that these types of discussion series could not happen without support and generosity from community members like you. We are a nonprofit research institute. And if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give or email our development team at development@btiscience.org. So thanks again, everybody, for your interest and your support of Boyce Thompson Institute. And thanks again, Lukas, for doing this with us today. It was great. I had a really, really good time with this discussion with you. And I hope everybody has a nice rest of your day.
Thanks. Bye-bye.
Bye-bye.